Hey everyone, Dr. D here, and in this video we're going to be covering Chapter 10, which deals with the cell cycle. So let's go ahead and get started. What is the cell cycle? You can think of it as the period of time from where a cell is made until that cell divides. So from the period of time from a cell is formed until that cell divides into two cells. And why do we need cell division? Well, obviously, you and I are multicellular creatures. We need cell division to grow from a fertilized egg called a zygote. One cell becomes two, becomes four, becomes eight, and our growth and development depends on the cell cycle. Also, you use the cell cycle for repair. If you are a prokaryotic cell, like an E. coli, for instance, or Salmonella, the cell cycle is simply for reproduction. Through asexual reproduction, one E. coli becomes two separate E. coli, whereas in our bodies, obviously, cell division is for repair, for growth, for development. Okay, so one thing you need to realize is, let me show you this slide first. One thing you need to realize is that there are two major types of cells in your body. There are what are known as somatic cells. These are the non-reproductive cells. And then there are what's known as the gamete cells. These are the reproductive cells, including the sperm and the eggs. So why do I point that out? Well, because, again, the gametes are the sperm cells and the egg cells in your body. Okay? And the cell division that results in the sperm and the eggs is cell division called meiosis, okay? And that we're going to cover in Chapter 11, not this chapter. This is Chapter 10. So just keep in mind that for the next chapter, we are going to be discussing meiosis, and that is a special type of cell division that can produce the sperm and the egg cells. And what are the sperm and the egg cells called? They're called the gamete cells, or the reproductive cells, okay? That's not the topic of this chapter. The topic of this chapter is how do all the rest of our cells, so every cell that's not a sperm cell in males and egg cell in females, all the rest of the cells, how do they divide? So we got skin cells, we've got brain cells, we've got liver cells and pancreas cells and stomach cells, and you've got so many different types of cells in your body. How do they divide? Those cells are called somatic cells. And how does a somatic cell divide to form two somatic cells? Okay, that's the topic of this chapter. That's the topic of mitosis. Okay, mitosis is what we're going to be covering now. Now, to understand mitosis, what you need to understand first is that you have a bunch of cells. We are a multicellular creature. We're made up of billions of cells. And for the most part, our cells contain a nucleus, and that nucleus contains all of our DNA, our whole genome. You know what genome means? Genome, here's the word here. The genome is your entire DNA. Right, it's all of the DNA that you possess, all of the instructions, the blueprints to make you. That's your genome, and each and every one of your nucleated cells contains your entire genome. So this skin cell here, that skin cell contains a nucleus. That nucleus contains all of the blueprints to make you again. Okay, your brain cells contain a nucleus. That each of those cells has your whole genome. Your skin cells have your whole genome. Your brain cells, your heart cells, your liver cells, your pancreas cells, every cell in your body has your whole genome, as long as it's a nucleated cell. Okay. Now, why do I say nucleated cell? Well, some cells in your body don't have a nucleus, and so they don't have a genome. Those are your red blood cells and your platelets. That's just a fun fact. You don't need to remember that. But yeah, just interesting to know that your red blood cells don't contain DNA. And the reason for that is as the red blood cell or erythrocyte is being formed, it actually squirts its DNA out. It squirts its nucleus out in an effort to be a more compact cell, so it can get between all of the little capillaries and into tight nooks and crannies where it can deliver oxygen. But you don't need to know any of that. That's just fun facts for you. So again, what's the genome? The genome is your entire subset of DNA, all of your chromosomes. So what's the difference then? Look at this. What's the difference between a genome and a chromosome? So what is the difference between a genome and a chromosome? Genome is all of your DNA. Chromosome is a DNA molecule. Okay, so imagine double-stranded DNA. Double-stranded DNA from beginning to end. One piece of double-stranded DNA from beginning to end. That's a chromosome. Okay, think of that as a chromosome. So a piece of double-stranded DNA, one molecule of double-stranded DNA, that's a chromosome. So how many pieces of DNA do humans have? You and me. How many a typical human inside of their nucleus of each and every one of their nucleated cells? How many chromosomes are inside? How many pieces of double-stranded DNA inside of each and every one of your nucleated cells? Well, the answer is 46 chromosomes. Okay, 46 chromosomes is the number of chromosomes a human possesses. We have 46 pieces of DNA inside each and every one of our nucleated cells. Okay, good. So a genome is all of your chromosomes. A chromosome is a one molecule of double-stranded DNA. Now we know. That's great. That's great. So what are genes then? Before we move on, what are genes? What you need to know is that genes are the genetic information that codes for a particular protein. Genes code for proteins. And where are the genes in your genome? Well, they're on your chromosome. So if a chromosome is a double-strand DNA, each chromosome can contain the information for hundreds or thousands of genes. Okay. Another thing to realize before we move on is prokaryotes like E. coli or Salmonella, etc. Prokaryotes usually have only one chromosome, and it's not a linear double-stranded chromosome with two ends, right? It's usually a circularized double-stranded DNA chromosome. Okay, so you have one circularized chromosome. Good. So what else? So remember what we said before in chapter, I believe it was chapter 4. We said the chromosomes don't exist as simply DNA. The chromosomes exist as chromatin. Remember this term chromatin from before? Chromatin means DNA wrapped around protein. There were these special proteins called histone proteins, and the DNA wraps around the histone proteins, forming a nucleosome. This is the basis of chromatin. What does that mean? Look, here's your double-strand DNA, right? This is called naked DNA, double-stranded DNA. When you have chromatin, which means how your chromosomes actually exist, your chromosomes don't just exist as plain double-stranded DNA. No, your chromosomes exist as chromatin, which means, first of all, you take these proteins here. This here, what I'm highlighting or what I'm pointing out right now is the histone proteins. There's actually 8 of them, so it's called the octamer of histone proteins. This forms what's called the histone core. These 8 histone proteins form the histone core. It's a quaternary structure. And then the DNA, the double-strand DNA, wraps around that about approximately 2 times. Not exactly 2 times, but approximately 2 times. Now that is the basic unit of chromatin, and it's called the nucleosome. The nucleosome is 1 octamer of histone with DNA wrapped around. And when you have multiple of these histones, and these nucleosomes, I should say, these nucleosomes all kind of coil up into what looks to be an old school phone cord looking structure, right? That's called solenoid, or you could just think of that as chromatin. This is chromatin. Chromatin is DNA plus protein, plus histone proteins. And here's what you need to understand. This is how your DNA exists. Your chromosomes exist as chromatin. And 90% of the time, your DNA looks like this. This is really important to understand real quick. 90% of the time, your DNA looks like this. And I want to show you something real quick on our whiteboard. I want to show you something very important, and that's how DNA packages itself during cell division or before cell division. I just want to show you something actually quite important to understand, as it will make sense of a lot of this stuff that's to come. So I've got my whiteboard clean now. I'm going to grab a whiteboard marker, one of these dry erase markers, and I'm going to show you something. And that's this. Let's go back to that slide here. All right, I just want to show you something. If I have a cell, okay, if I have a cell, and that cell, is a eukaryotic cell, it's going to have a nucleus, okay? Okay, there's the cell, there's the nucleus. The chromosomes are inside of the nucleus, right? The chromosomes are inside of the nucleus. But here's the thing that you need to understand. The DNA is very loose 90% of the cell cycle. 90% of the time, the DNA inside of the nucleus is quite loose, like spaghetti. So, if I were to look inside of the cell and look at the DNA 90% of the time looks like a bowl of spaghetti. I can't see individual chromosomes. I can't tell there's 46 chromosomes. If I look inside of the nucleus, if I look at the DNA, it just looks like strings or spaghetti, okay? The only time the DNA starts to condense so that I can actually tell there's 46 different chromosomes is right before mitosis, right before the cell divides, okay? So for example, when my skin cell is just being a skin cell and just doing its job, there's really no need for the DNA to be all condensed. The DNA is all unraveled. It's like, I call it spaghettified. It's what's called euchromatin. The DNA is just kind of unraveled. And that DNA is easy to get to. It's easy to read. That way your enzymes can get in there and read the genes and make the proteins from those genes. Okay? But let's say the skin cell decides to divide, okay? So now my cell wants to divide. What do you think I should do with my bowl of spaghetti DNA? Like, I've got all this DNA just floating around like a bowl of spaghetti. Do you think that's convenient for a cell that's going to divide? Probably not. So, the first thing I got to do is copy all the chromosomes, but I'm not talking about that part. What do I have to do with the DNA before I actually split the cell into two cells? I'm going to split the cell into two cells. Well, that DNA should condense, right? So that DNA, each of those chromosomes starts to condense and condense and condense, and pretty soon you start forming these tight clusters like this, you see? Your chromosomes condense and they become kind of visible. And that's because the chromosomes kind of coil up. They condense. Now look at this. Let me show you something. Again, see this image here at the bottom right? Those are nucleosomes forming chromatin. Again, this is your bowl of spaghetti. This is when the DNA is loosely packed. Why would the DNA want to be loosely packed like this? Well, because this, loose DNA is very easy to read. It's easy to get to the genes and read the genes so you can make proteins. But then when the cell's dividing, you're not really reading the genes, are you? You're focused on dividing the cell. So you know what the chromatin does? It forms these things called chromatin loops. Those chromatin loops form these things called chromatin rosettes. Right? Rosettes of chromatin loops. And then those rosettes actually fold further, and then you've got this really tightly coiled chromatin called the mitotic chromosome. Did you follow that? So your DNA forms these structures. This is what you may think of when you think of chromosomes. You know, when you've thought of chromosomes in the past, or you've seen cartoons of chromosomes or textbook image of chromosomes, you've seen this structure, haven't you? It looks really, really condensed like this. It's obviously not just a bowl of spaghetti, is it? Well, what I want to tell you is that your DNA only looks like this 10% of the time. And that 10% of the time is pretty much during mitosis, when the cell is actually dividing. When one cell is dividing into two, that's the only time your DNA looks like this. Otherwise, your DNA starts to uncoil, 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 back to like a bowl of spaghetti. And there's no way you're going to see individual chromosomes when it's a bowl of spaghetti. So I just want you to understand that. The DNA is loosely packed. It's actually not even packed. It's just unraveled. And then when the cell wants to divide into two, it starts coiling, or I should say condensing up, condensing, forming these chromatin loops, these rosettes, and the mitotic chromosome. So when I say mitotic chromosome, I mean the condensed chromosome. When I say regular chromatin, I mean the uncondensed chromosome, the non-mitotic chromosome. Okay, so that's good to understand. All right, what else do we need to understand? So let's talk about humans and how many chromosomes we have. Remember I mentioned earlier we have 46 total chromosomes. Humans have 46 total chromosomes. But we're said to have two sets of chromosomes. What do I mean? Let me show you. Here are the chromosomes from a human. This is what's known as a karyotype. It's an actual laboratory procedure where you could take mitotic chromosomes from a cell nucleus and line them up. Okay, so you'll see you have these two are called chromosome 1. You have two chromosome 2s. You have two chromosome 3s, 4, 5, and so on and so forth. You have 23 chromosomes, 23 different chromosomes, but you have two chromosome 1s, two chromosome 2s, two chromosome 3s, two chromosome 4s. This is really interesting. So this is showing you, again, the karyotype of a human genome. This is showing you all of the mitotic chromosomes in order from a cell, from a nucleated cell. And so what do we notice? We notice that even though there's 46 total chromosomes, if you count 1, 2, 3, 4, 5, 6, if you count all of these individual chromosomes, you would arrive at the number 46. However, look what's going on. You have two chromosome 1s. You have two chromosome 2s. You have two chromosome 3s. What's that all about? Well, humans have 46 chromosomes, but they actually, it's more accurate to say that we have two sets of chromosomes. We have two sets of 23. A set means chromosomes 1, 2, 3, 4 through 23. We have 1 through 23. That's a set of chromosomes. And humans have two sets of 23. We have two complete sets of chromosomes. And a complete set of chromosomes, again, is chromosomes 1 through 23. Okay, chromosome 1 is the longest chromosome, chromosome 2 is the second longest, chromosome 3 is the third, and so on and so forth until you get to chromosome 23. And in fact, chromosome 23 is called the sex chromosomes. Why? Because they determine your sex. This is the X chromosome, and this shorter one is called the Y chromosome. And male animals have an X and a Y chromosome, and females have two X chromosomes. Okay, and humans, an X and a Y is male, and two X's would be female. So this would be from a male cell. Okay. The rest of these chromosomes are called autosomal chromosomes. Now, why do you think we have two chromosome 1s, two chromosome 2s, two chromosome 3s, two chromosome 4s? Why do we have two of every chromosome? Well, let's think about that. Where did you get your chromosomes from? Where did you get your genetic material from? You got your chromosomes from your parents. Does that make sense? These chromosomes are from your parents. So you inherited one set of chromosomes from your mom, and you inherited another set from your dad. So look at this. You see how I have 2 chromosome 1s? Well, one of these 2 is from your mom. One of these 2 is from your dad. Same thing with these 2 chromosome 2s. One of these chromosome 2s is from your mom, and one of these chromosome 2s is from your dad, and so on and so forth. You inherit one set of 1 through 23 from your mom. You inherit one set of 1 through 23 from your dad. And that's how you get your 2 sets of chromosomes. The sperm has 23 chromosomes. The egg has 23 chromosomes. When the sperm from your dad meets the egg from your mom, that's how you get 2 sets of chromosomes. 2 chromosome 1s, 2 chromosome 2s, 2 chromosome 3s. Okay. So, the name for that, before I forget to, sometimes I forget to point this out, the name for that is called diploid. Humans are diploid. Another way of saying diploid or denoting diploid is 2n. What does that mean? You have 2 complete sets of chromosomes. 2 complete sets of chromosomes for 46 total chromosomes. Where did you get each set? You got 1 set from your mom from her egg. You got 1 set from your dad from his sperm. Okay? So diploid organisms are those with 2 complete sets. A maternal set from mom and a paternal set from dad. Okay? Haploid organisms, on the other hand, they do not have complete multiple sets of chromosomes. They only have 1 set of chromosomes. So, for example, your sperm cells only have 23 chromosomes. Your sperm cells are called haploid. Your egg cells only have 23 chromosomes, and your egg cells are called haploid. But when the sperm meets the egg, now it's called diploid. From that point on, it's diploid. Your organism is diploid. Okay. So what about think about this one. What about a bacterial cell? So if I have like an E. coli cell, remember E. coli, how many chromosomes do they have? E. coli only has 1 chromosome. Remember, prokaryotes usually only have 1 chromosome, and it's a circularized chromosome. So do you think E. coli cell is diploid or haploid? Haploid. It's haploid. So prokaryotes are typically haploid, whereas eukaryotes are typically diploid, okay? Because eukaryotes usually have a mom and a dad, usually. Okay. Not always, of course. What else? So I have a mom and a dad chromosome 1, a mom and a dad chromosome 2, a mom and a dad chromosome 3. So let me ask you something. Do you think this chromosome 1 from your mom is identical to this chromosome 1 from your dad? Think about that for a second. Would chromosome 1 from your mom be identical to chromosome 1 from the dad? So if you were to look at the A's, G's, C's, and T's, the actual genetic sequence of these chromosomes, would your dad's chromosomes say the exact same thing as your mom's chromosomes? Would this chromosome 1 from your mom say the exact same stuff as this chromosome 1 from your dad? Well, I hope not, right? That would mean they're related, and not just related, they're like twins or something, which actually is impossible because they'd have to be identical twins, so they couldn't be of opposite sexes. But anyway, this chromosome 1 is not the exact information as this chromosome 1. Does that make sense? This chromosome 1's from your mom, this chromosome 1's from your dad, and your mom and your dad obviously have different genetics. They're not they're not twins. They've obviously got different genetics going on, okay? So then you might ask, well, Dr. D, if this chromosome 1 from my mom is not the same as this chromosome 1 from my dad, why are they both called chromosome 1? What do they share in common besides the fact that they're both the longest chromosome, right? Well, they do share something in common. I want to tell you what they share in common. What the 2 chromosome 1s, the 2 chromosome 2s, the 2 chromosome 3s, what they share in common is the types of genes found on those chromosomes. Do you follow that? The types of genes found on the chromosome numbers matches. So, for example, I'm just going to give you some random examples. They're not even accurate because I don't have time to look it up right now. But let's just pretend, just for pretend. Let's say eye color. I was interested in eye color. Let's pretend. Well, I know that eye color gene is on chromosome 7. Does that make sense? So if this is chromosome 7 from your mom, eye color gene might be right here at this spot on chromosome 7 from your mom, which would mean it would be at the similar spot on chromosome 7 from your dad. Does that make sense? So again, I don't remember which chromosome eye color is on because I could easily look that up, and that's not the point of what I'm trying to tell you. What I'm trying to say is, for example, if eye color is on chromosome 7 for me, it's on chromosome 7 for you, it's on chromosome 7 for your husband or your wife or whoever, your cousins, your uncles, your aunts, every human has eye color information on chromosome 7, for example. So eye color information will be on chromosome 7, whereas I don't know, hair color information might be on chromosome 12. Again, I haven't looked these up right now, but I'm just giving you an example. If eye color gene is on chromosome 7, it's on everyone's chromosome 7. If hair color gene is on chromosome 12, it's on everyone's chromosome 12. So the difference between the chromosome numbers is simply what types of genes are on them. But does that mean you have the same exact information? No. So for example, your mom might have blue eyes, and your mom might have given you the gene for blue eyes right here on mom's chromosome 7. But here on dad's chromosome 7, your dad may have given you the gene for brown eyes, right? So, because he has brown eyes. So here would be a brown eye chromosome 7. Here would be a blue eye chromosome 7. They're both eye color genes, but they don't have to say the exact same thing, okay? That's the difference between these two different sets of chromosomes, the set from mom and the set from dad. The types of information are the same, but the exact instructions are different, okay? Now, here's something you need to understand. You know how I've been saying mom and dad's chromosome 1, mom and dad's chromosome 2, mom and dad's chromosome 3? Instead of saying that, there's actually a specific term for mom and dad's chromosome 1. They're called homologous chromosomes, okay? So these two are homologous chromosomes. So when I say homologous chromosomes, just remember, I mean mom and dad's chromosome 1, mom and dad's chromosome 2, mom and dad's chromosome 3. That's what homologous chromosomes means. It means the maternal, mom's, and paternal set of chromosomes. So humans have homologous chromosomes inside of each of their nucleated cells. Humans have homologous sets of chromosomes. One set of 1 through 23 is from mom. One set of 1 through 23 is from dad. So you have two sets of chromosomes, okay? Two homologous sets of chromosomes. So let me show you this real quick. Here. Look, if this is chromosome 6 from your dad, okay, if this cartoon represents chromosome 6 from your dad, this cartoon represents chromosome 6 from your mom. Again, are these identical in information? If you looked at the actual genetic information, does this say the exact same stuff? No, remember, this one's from your mom, this one's from your dad, this one might say one type of thing, this one says another, but the types of genes are the same. The types of genes are the same. If this one has hair color, that one also has hair color. If this one has eye color, this one also talks about eye color, but it doesn't mean it's the same exact color, okay? So anyway, these are called homologous chromosomes, the maternal and paternal set of chromosomes. Okay, so that's what homologous chromosomes mean. Just remember, homologous chromosomes are not identical. They are the maternal and paternal set of chromosomes, okay? Now, what if a cell, like a skin cell, what if a cell wants to divide into two cells? Okay, what if a cell wants to divide into two cells? What does that cell have to do with its chromosomes ahead of time? If you think about it, you know, when a cell divides, like a skin cell divides into two, it wants each of those daughter cells to have the same genetic information. Does that make sense? Like if one of your skin cells is going to divide, you want to make sure that both of those new skin cells have the same information as each other and the same information as their parent cell, right? So how would you do that? Look at the 46 chromosomes. What would you have to do first before you divide the cell? Well, you have to copy all of these chromosomes. Does that make sense? You see how you have 46 chromosomes? Well, before the cell divides, guess what? You have to copy each and every chromosome. So you have to make 2 copies of this chromosome. You have to make 2 copies of this chromosome. You have to make 2 copies of this one, 2 copies of this one, 2 copies of this one. So your 46 chromosomes effectively becomes how many chromosomes? Well, 46 times 2 is 92. 92 total chromosomes, right? You're going to make 92 chromosomes because now you've taken your homologous chromosomes and you've copied every single chromosome. Look what you've done. So right before the cell divides, remember you had homologous chromosomes, but look, remember if this was chromosome 6 from your dad, right before the cell divides, you got to copy it, right? You see, you got to copy this. And now look what you have. Now you've got 2 chromosome 6s from your dad. These are literally identical carbon copies, okay? These are identical copies. And you did the same thing with mom's chromosome 6, didn't you? You took mom's chromosome 6, you copied it. Now you have 2 exact copies of mom's chromosome 6. So, remember the blue and the red were homologous chromosomes? Well, what are the 2 blues called, or the 2 reds called? What are they called? Those are called sister chromatids. Look at this. Sister chromatids means identical copies of a chromosome. Sister chromatids are identical. Identical in every way. You know how they could be identical in every way? Well, because all you did was take a chromosome and copy it. Word for word, letter by letter, right? Okay. So, sister chromatids are identical copies of every chromosome. Homologous chromosomes are the maternal and paternal sets. They're not identical. They just have the same types of information on them. If you want more clarification, you want another video explaining that in more detail, click here. I go into even more detail as to what a homologous chromosome is versus a sister chromatid. And why am I stressing this so much? I'm stressing this because the better you understand homologous chromosomes versus sister chromatids, the easier Chapter 10, covering mitosis, and Chapter 11, covering meiosis, are going to be. Those chapters are going to be super easy once you understand the difference between these terms. That's why I'm harping on these so much, and I really want you to understand this. Now, before I take a little break, I want you to notice that these are mitotic chromosomes. Remember, mitotic chromosomes means the chromosome after it's like very condensed. Very condensed because the cell is about to divide, about to undergo the process of mitosis. What you need to understand is the mitotic chromosomes have this narrow portion. You could think of it as a narrow waist, okay? That narrow portion, narrow waist of each mitotic chromosome has a name. It's called the centromere. So you should know that. The centromere is the narrow portion. You could think of it as the narrow waist of the mitotic chromosome, the chromosome once it's condensed. Okay? And once you have sister chromatids, remember identical copies of chromosomes, before the cell divides, these sisters find each other and they snap to one another. The sister chromatids attach to one another at the centromere. And the proteins that hold the centromeres together are called the cohesin proteins. The cohesin proteins hold the centromeres to one another. That's why in those cartoons or from your old textbooks from high school or junior high, you know that the chromosomes look like X's, little X-shaped structures. Well, it's because you've got the sisters held together at the centromere by cohesin proteins, okay? So these are all terms to understand. Homologous chromosomes versus sister chromatid pairs, centromeres, cohesin proteins. Now, what's this other protein on the outside of the centromere? It's called the kinetochore. It's actually a little complex of proteins called the kinetochore. So what's the kinetochore do? What is that? It looks like a little jelly bean in this cartoon, right? What is that? Well, you don't need to understand this at the moment, but you will in a little bit. But let me foreshadow. Look at this. These are sister chromatid pairs held together by cohesin. There's the kinetochore jelly bean shaped looking thing. Look what the kinetochore can do. What are these? Does it even say what these are? Microtubules. Microtubules. Remember the cytoskeletal component that we talked about in chapter 4 called microtubules? Here's the thing you need to understand. The kinetochores on the side of the waist, the kinetochores are docking sites. The kinetochores are docking sites for microtubules. When these microtubules get close, the microtubules can dock, can attach to the kinetochore. Okay? So I'll show you why this is important later. I'll show you why this is important. Okay? So good. Oh, and here's another fancy terminology. Okay, ready? Look at this. What was this blue? The blue, if this was, I don't know, chromosome 6 from the dad, this chromosome is homologous to that chromosome, right? This chromosome 6 from the dad is homologous to that chromosome 6 from the mom, okay? But what happens after DNA replication? You've got sister chromatids from the dad, sister chromatids from the mom, and you ready for the fancy part? Here's another thing you could say. You could say this pair of sister chromatids, you see these two blue ones, this pair of sister chromatids is homologous to this pair of sister chromatids. Did you find that? Did you figure that out? This pair of sister chromatids, these two chromosome 6s from dad, is homologous to this pair of sister chromatids. These two, for example, chromosome 6s from the mom. Okay, this is the paternal sister chromatid pair. This is the homologous maternal sister chromatid pair. So hopefully that terminology helps you. Again, the better you understand all this terminology at the moment, the better you're going to understand all of these concepts. And that's a fact. So with that, let me take a quick little break, and I'll be right back. So break time. Hey everyone, Dr. D here, back from break. I was about to tell you about the cell cycle in more detail. So let's go ahead and get started. The cell cycle consists of two phases, two major phases. These are called the mitotic or M phase and interphase. All right, mitotic phase and interphase. Real quick, interphase, let me tell you about interphase first. Interphase takes up roughly 90% on average of the cell cycle time, and it can be divided into 3 subphases. So interphase, 90% of the cell cycle can be divided into subphases. Those subphases are called G1 or growth or gap 1, S phase for synthesis, and G2 for gap phase or growth phase 2. Okay, what's interphase? Interphase, again, it takes up 90% of the cell cycle. And interphase, you can think of it like this. Interphase is the period of time where the cell is being a cell, and the cell is growing, it's doing its job being a cell. During interphase, the cell is just doing its job. So if it's a skin cell, it's doing its skin cell duties. If it's a liver cell, it's doing its liver cell duties. It's just being a cell. Okay, and not only that, but during interphase, you can also prepare for cell division. So during interphase, the cell is doing its job, or it's preparing for division, preparing to divide. Okay, so that takes a long time, 90% on average of the cell cycle. Now, once the cell does decide to divide, it proceeds along these subphases of interphase and then enters mitotic phase. Mitotic phase, or M phase, is the other 10% roughly of the cell cycle. And this is where the cell's actually dividing. You can think of mitotic phase as the phase where the cell's dividing. Okay, and the M phase consists of mitosis and cytokinesis. Okay, mitosis and cytokinesis together make up M phase or mitotic phase. What you should realize is mitosis is not mitotic phase. Mitosis plus cytokinesis together is mitotic phase or M phase. So again, the two major parts, the two major components of the cell cycle are interphase followed by mitotic or M phase. Okay, and remember, interphase, it's when the cell's being a cell or preparing to divide. It takes about 90% of the cell cycle time, broken up into subphases G1, S, and G2. Okay. Mitosis, on the other hand, that's when that's part of M phase, and cytokinesis is part of M phase. So what's the difference between mitosis and cytokinesis? They're both part of M phase, but what is mitosis? Let's see if I have a good definition. Mitosis is the division of the genetic material in the nucleus. Did you follow that? Mitosis is when the DNA separates. Okay, you could think of it like the sister chromatids separate from one another. The DNA separates that to give you two genetically identical cells. That's mitosis. So what's cytokinesis? Cytokinesis is when the cytoplasm separates. So the actual cell separates into two cells. That's cytokinesis. When the sister chromatids separate from one another, when the DNA separates into two cells, that's mitosis. So mitosis is separation or division of the genetic material. Cytokinesis is separation or division of the cytoplasm. Okay, follow it that way. And together they comprise M phase or mitotic phase. Okay, mitosis itself is broken up into 5 subphases itself. Those phases are prophase, prometaphase, metaphase, anaphase, and telophase. Okay, prophase, prometaphase, metaphase, anaphase, and telophase. Okay, those are the subphases of mitosis. Okay, and then we're going to talk about cytokinesis as well. Okay, good. So you could see all of the phases here, okay? Remember, the cell cycle is broken into two major phases, interphase and M phase. Interphase has the subphases G1, S, and G2. See? G1, S, and G2. That's the interphase. Followed by M phase, which consists of mitosis. The purple arrow is mitosis. Prophase, prometaphase, metaphase, anaphase, telophase. Those are subphases of mitosis. And then you got cytokinesis, which is the blue arrow here. The blue arrow here is cytokinesis. Okay? So these arrows represent the cell cycle. And notice how M phase is only roughly about 10% of that time. Okay? So, how does it work? Here is interphase. And by the way, I have really good videos here going into even more details. At the bottom left, you see I have a video down here at the bottom left of interphase. I have a video for prophase, prometaphase. I have a video for metaphase, anaphase, telophase, and this video here, let me show you. This video here is a great review of all of it. So I'm going to go through it a little bit quickly in this video, but if you want even more details, you want extra details, watch these videos on these slides, because I do have a lot of extra videos already posted for you that go into much more detail, more than I'm going to go through in this video. So please do watch those if you're at all confused, but I will cover it. So interphase, remember interphase is the period of time where you have 3 subphases, G1, S, and G2. And remember I said this is when the cell is preparing to divide or just being a cell. So during the first gap phase, G1, cells undergo major proportion of growth. So the cells just grow. What do we need to know about G1? No, G1, the cells are growing. Then you enter S phase, synthesis phase. What happens during synthesis? What are you synthesizing? DNA. You're copying all the chromosomes. Remember when I said humans have 46 chromosomes and then you copy all the chromosomes, you have 92 chromosomes? That's happening during S phase. So after S phase, guess what you have? Remember this? Look, all of your chromosomes replicated, and now you have copies of all the chromosomes. So only at this point do you have sister chromatid pairs. Does that make sense? Only after, I'm sorry, I'm trying to get to the slide. Only after S phase do you have sister chromatid pairs. Then you have G2, gap 2, or growth phase 2. This is where the chromosomes start to coil up. Do you remember what I said earlier? I made a big point that your DNA is very loose, like a bowl of spaghetti. That chromatin is very loose. However, when the cell is about to actually divide, which means M phase, mitotic phase, the DNA starts to condense and condense and condense into those mitotic chromosomes that look like X's, right? So that condensation of the chromosomes, that coiling up of the chromosomes, starts at G2. Also, do you remember this structure we talked about in chapter 4? This structure I'm indicating right now, those are called the centrioles. Remember, animal cells have centrioles? The centrioles actually replicate. So now you have 2 pairs of centrioles. That also happens during G2. I'll explain why that's important later. So again, interphase, 90% of the cell cycle. The cells undergo 3 subphases, G1, S, and G2. Please note that during G1, the cell grows. During S, the DNA gets replicated. And during G2, the DNA starts to condense and the centrioles replicate. All right, now look at the nucleus. Look inside of the nucleus. Do you see individual chromosomes in the nucleus? Do you see all the little chromosomes, the mitotic chromosomes in there? The answer is no. You know why? Because the DNA is strewn about. It's loosely packed. It looks like a bowl of spaghetti in there. You can't see individual chromosomes at this point. Okay, so again, you want more information, you want to watch this in more detail, go ahead and click this icon here on your PDF notes at the bottom left. Okay, so prophase. By the end of interphase, right, look at this. By the end of interphase, you've undergone G1, growth, S, synthesis, and G2, centrioles replicated, DNA condensed. And now you're ready to enter M phase, right, which starts with prophase. So now we're ready to enter mitotic phase. So let's talk about that. During M phase, the first part is called prophase. And what happens during prophase? Well, if you look here, it tells you the bullet points are very important to understand. The DNA actually condenses. And look what's formed. What does the DNA exist as? Look at the chromosomes. What are these? What are these? They're X-shaped structures, aren't they? And what are those X-shaped structures here? Well, aren't those the sister chromatid pairs that have paired up, right? The sister chromatid pairs that we talked about before. So remember this slide here, sister chromatid pair, sister chromatid pair. So those sister chromatid pairs pair up. Remember with the cohesin proteins holding the sisters together at the centromeres, right? And why do they look like little X's? Why can you see individual chromosomes at this point? Well, because of the DNA has condensed into those mitotic chromosomes, right? What you need to know is that the nucleus is still intact. The nucleus is still there. See the nuclear envelope, the double membrane around the DNA? It's still there, but it's breaking apart. It's starting to break apart. The ER and the Golgi break down into vesicles. Another thing that happens is you see these centrioles. You see these centrioles here? These centrioles start moving to this end of the cell. This pair of centrioles starts to move to the other end of the cell. And do you also see these green things coming out from the centrioles? It looks like a spider web. This little spider web. It's actually called the mitotic spindle. You know what the mitotic spindle means? It means that microtubules, you remember what microtubules are, it's a cytoskeletal component made of alpha and beta tubulin. Microtubules start to synthesize, start to form from the centrioles. The centrioles are what is known as a microtubule organizing center. Microtubules organize themselves and grow out from there. And I'll explain why this is important soon. Just know that the centrioles start drifting apart, moving to opposite poles of the cell, and mitotic spindles start to form. Microtubules start to grow out from them. Okay, that's what we need to know. And again, you want to watch this in more detail, watch this video here on Prophase. Prometaphase, what's going on? This centriole bundle is here at one pole of the cell. This centriole bundle is at the other pole of the cell. And at this point, look at this. At this point, those centrioles are actually called centrosomes. You know why? You know what centrosome means? Centrosome means the centrioles plus the mitotic spindle. Centrioles plus the mitotic spindle. Once they are at opposite poles of the cell, you stop simply referring to them as centrioles and you start calling them centrosomes, okay? Not to be confused with centromeres, which is the narrow waist of the mitotic chromosomes. They're centrosomes, which means centrioles plus microtubules, the mitotic spindle. So anyway, during prometaphase, which is the second subphase of mitosis, is the nucleus still there? Remember the nucleus? Is the nucleus still there? Nope, the nucleus has broken down. The centrosomes are at opposite poles of the cell, and look at these look at the mitotic spindle. Look at the microtubules. They've really grown, right? You see those microtubules? They've really grown. And look what the microtubules have done. Can we zoom in on this at all? Let me try something. I wanna zoom in on this. I wonder if I can. Oh, maybe I can show you. Let me zoom in on this a little bit. Look, this is actually really neat. Let me show you something. Look here. Look here. Look what the mitotic spindle is doing. The microtubule is getting close to the kinetochore. Remember the proteins at the centromere? And they're attaching to one another. The microtubules are attaching to the kinetochore at the centromere of the mitotic chromosome. So look at that, where my dot is. The microtubules from this centrosome, microtubules attaching to the sister chromatid pair at the kinetochore. Remember this drawing here? Remember what I said these jelly bean structures are? They're called kinetochore. The kinetochore are docking sites for those microtubules. Microtubules can attach to kinetochores. And where are these microtubules coming from? They're coming from the centrosome on this end, which attaches to this kinetochore on this sister chromatid, and the centrosome on that end, where the microtubules attach to this kinetochore on the other sister chromatid. So do you see what happened there? During prometaphase, microtubules from the centrosomes are attaching to kinetochore on opposite sides, right? On opposite sides of the sister chromatid pair. And at this point, microtubules are attaching to the sister chromatid pairs. And do you see these pairs, these sister chromatid pairs? They start making their way to the center of the cell. They start making their way to the center of the cell. Have they made it to the center of the cell? No, but they're making their way to the center of the cell. So look at metaphase. This is the next subphase, metaphase. Notice how the sister chromatid pairs have lined up at the center of the cell. Sister chromatid pairs have lined up at the center of the cell, and each sister is connected at the kinetochore to a microtubule from opposite centrosomes, right? Centrosomes from opposite poles of the cell. So remember, these sister chromatid pairs are identical copies. So for example, if this is chromosome 1 from the mom, right next to it is the sister, which is another exact copy of chromosome 1 from the mom. If this is chromosome 1 from the dad, it's attached to the exact same copy of chromosome 1 from the dad. So what you could say is the information's lined up, and each pole of the cell is attached to one copy of all the information. Okay, that's metaphase. And this imaginary line, you see this imaginary plate. They call this the metaphase plate. It's an imaginary plate on which the sister chromatids line up. And by the way, this is a very simplified version of a cell, isn't it? You see how there are 1, 2, 3, 4 sister chromatid pairs? Okay. In a human, there would be a lot of more of these pairs. There would actually be 46 of these pairs. Okay, 46 of these X's would be lined up at the center of the cell during metaphase. This is a simplified image. Here, there's only 4 pairs lined up. So, for example, this is 2 copies of mom's chromosome 1. This is 2 copies of dad's chromosome 1. This here would be 2 copies of dad's chromosome 2. This would be 2 copies of mom's chromosome 2. So you're only showing chromosomes 1 and 2. Humans would have chromosomes, they would have 46 of these because you would have 2 mom's chromosome 1, 2 dad's chromosome 1, 2 dad's chromosome 2, 2 mom's chromosome 2, and you would have, 46 different X's, 46 different sisters lined up. Just in case you're wondering. Then what? Do you remember? Okay, what's going to happen next? Look, do you remember the little cohesin proteins holding the sisters together? We talked about that earlier. See the cohesin proteins holding the sister chromatids to one another at the centromere, the narrow waist? Well, those break apart. Okay, the cohesin proteins let go, and now the sisters are separated from one another. So the sister chromatids are separated from one another. Did you see what happened during anaphase? Sister chromatids separate from one another, and they start moving to opposite poles of the cell. You see, sisters separate from one another. They move towards opposite poles of the cell, which is good because the same information is heading to the left as is heading to the right. Because remember, those sisters were just identical copies of the same information, right? So if they break apart and move to opposite poles of the cell, that means the same information is heading to one cell as is heading to the other cell. So that's good because what you need to realize is that mitosis results in 2 genetically identical cells. Did you follow that? The purpose of mitosis, the point of mitosis is to result in 2 genetically identical cells. They need to be genetically identical to one another, and they need to be genetically identical to their parent cell. Okay? How do you accomplish that? Well, why not take all 46 chromosomes, copy each and every chromosome, and then separate those copies, which is what you're seeing here. During anaphase, the copies are separating from one another. Okay, the sister chromatids, the identical information separating. Okay, and again, if you want to see this in more detail, you can watch this video at the bottom left. Okay. Oh, and by the way, before I finish with telophase, let me show you something. Some people ask, how are the sisters separating from one another during anaphase, what we just talked about? How are those sisters traveling apart? Are they being pulled apart, or are they making their own way apart? And look at this. Look at this real quick. Let me show you. If this is the centromere, this is the narrow part of the chromosome. This is the chromosome. This is the sister chromatid, you see? This is the kinetochore protein. Look what they have inside of the kinetochore protein. It's a motor protein. Remember these little motor proteins, kind of like dynein, which was in the flagella or cilia? These little motor proteins can walk. With the help of ATP, they can walk. And the kinetochore proteins on the side of the sister chromatid pairs, they contain little motor molecules which walk along microtubules. So to answer that question, why do the sister chromatids move to opposite poles of the cell? What you need to know, look, during anaphase, why are these sisters moving to opposite poles of the cell? Well, because their kinetochores contain motor proteins that walk them along the microtubules to the opposite poles of the cell. That's why. So just in case you wanted to know how that works, I may have a question on that on the exam. Okay, and then what the final phase, the final subphase of mitosis is called telophase. So what's happening during telophase? The sister chromatids have fully separated to opposite poles of the cell. the nucleus, see the nucleus starts to reform. Remember the double membrane of the nuclear envelope? The nucleus starts to reform here to form a daughter nuclei. Another nucleus starts to form here to form another daughter nuclei. The DNA starts to decondense. Remember the mitotic chromosomes at this point, they've separated so they can start to relax and uncoil. The Golgi and the ER start to reform. Okay, and that's the end of mitosis. But that's not the end of M phase. Remember, you also have cytokinesis to deal with. And before I talk about cytokinesis, remember this video is really good on this slide. If you want to see an entire beginning to end review of in detail of what I just said about cell cycle, you can watch this video. I go into a lot more detail with it here. Okay, so cytokinesis is part of M phase. It's part of this actual cell division, but it's the part where the cytoplasm separates, right? Cleavage of the cell into equal halves. In animal cells accomplish cytokinesis by forming a cleavage furrow. Plants form a cell plate. So what's the difference between a cleavage furrow in animal cells and a cell plate in plant cells? Let's talk about this. Here's the animal cell on the left, and then you got the plant cell on the right. Let's start with the animal cell on the left. Animal cells, remember animal cells do not have a cell wall. They just have a cell membrane. And so what do you do with the cell membrane? You start to constrict or pinch the cell membrane at the middle of the cell. This pinching of the cell membrane is called the cleavage furrow. Okay, basically you pinch the membrane until you've pinched the cell into two halves. Okay, and what you need to know for the exam, what you need to understand is that the reason you're getting this pinching here is because of the contractile ring of the microfilaments. So do you remember in chapter 4 we talked about microfilaments? Remember actin microfilaments? Those actin microfilaments are responsible for cleavage furrow formation. I think we even talked about that in chapter 4. We talked about how microfilaments play a role in cell division of the cytoplasm and cleavage furrow formation. Well, this is what we were talking about. So you should know animal cells, cytokinesis occurs by cleavage furrow formation, and what's responsible for that is actin microfilaments. Okay, whereas in plant cells can't just pinch in half, right? Like a plant cell has a cell wall that's very tough, and that cell wall is not just going to pinch in half. So you got a different strategy for cytokinesis in a plant cell. In a plant cell, what you do, vesicles, you know what vesicles are, right? Little phospholipid bilayer balls. Vesicles full of cell wall material from the Golgi line up. And remember what cell wall material mean? Remember what cell wall is made out of? The cell wall is made out of cellulose. So you can think of it as vesicles full of cellulose line up at the center of the cell. Those vesicles fuse to one another. Because remember what I said, when a vesicle meets another vesicle, it's kind of like soap bubbles interacting. They can form one big bubble. So imagine if I have a vesicle full of, this vesicle is full of cellulose. This vesicle is full of cellulose. When they touch, they form a bigger vesicle full of cellulose. Well, you see, vesicles full of cellulose from the Golgi line up down the center of the cell. They fuse to one another, forming this rudimentary cell wall called the cell plate. So you should know this is called the cell plate. And then the cell plate matures into a new cell wall. And so basically, you've built a new cell wall at the center of the cell. Whereas in animals, you've pinched the cell in half. Okay, so again, animal cells divide by cleavage furrow formation, facilitated by microfilaments. Plant cells divide by cell plate formation, facilitated by vesicles from the Golgi, full of cell wall material, cellulose. Okay, that's how you do it. And here's just mitosis in a plant cell, just to show you what prophase, prometaphase, metaphase, anaphase, and telophase look like. Notice that during telophase, not only has the DNA fully separated to opposite poles of the cell, daughter nuclei are forming, but a cell plate's also forming. What does that tell you? That tells you that cytokinesis can actually begin before mitosis is even finished. Does that make sense? So let's say you're getting out of anaphase and you're heading into telophase here. Before telophase is even over, cytokinesis can begin. So there's some overlap there. It's concurrent, right? There's concurrency in those processes. But what you need to know is mitosis is not cytokinesis. Together they are M phase or mitotic phase, but mitosis is its own thing. Mitosis means separation of the genetic material resulting in two genetically identical cells. Cytokinesis means separation of the actual cytoplasm or separation of the actual cell into two cells. Okay, they can happen concurrently, but they are not the same thing. Okay, this is just showing you those vesicles I told you about, full of cell wall material from the Golgi, those vesicles fusing to one another to form a new cell plate. Okay, so what else do we have? All of this stuff is review of what we talked about. By the way, you've seen this term aster. I don't think I asked you about this, but the term aster is referring to the shorter microtubules that are coming off the centriole, or I should call it the centrosome. These short microtubules that are going towards the plasma membrane. It looks like a little star, doesn't it? It looks like a little star, and aster means star. So when someone's referring to the aster structure, they're talking about these short microtubules. I don't think you really have to know that, but just for your information. Okay, so let me take a quick little break. I'll explain binary fission in bacteria. Then we'll talk a little bit about the cell cycle control system, and that'll wrap up this chapter, chapter 10. So let me take a quick little break. Maybe grab a glass of water. I'll be right back to explain the rest of these slides and finish up chapter 10 for you. All right, break time. All right, welcome back, everyone. This is it, the last part of chapter 10. We're about to talk about binary fission. You can think of binary fission as cell division in prokaryotes. So bacteria, archaea, how are they dividing? That process is called binary fission. Bacteria do not divide. Prokaryotes do not divide by mitosis. They divide by binary fission. Binary meaning two, fission means to split into two. So you've got asexual reproduction going on during binary fission. Okay, remember something important. You have a single circular bacterial chromosome. Remember, you've got one double-stranded DNA and it's a circle. Okay, and in that circle somewhere, you've got what's called the origin of replication. Let me show you. So here's the bacterial cell, for instance, the prokaryotic cell. You got the cytoplasm inside, and then you see this circle. This circle represents the circular chromosome, right? Circularized DNA, double-strand DNA. Now, what happens during binary fission? Well, the first thing that happens is on one particular spot along that circle, on one particular spot, that spot's called the origin of replication, the DNA starts to get copied. And it gets copied in both directions at the same time. You see what's happening here? DNA is being copied toward the left and toward the right at the same time. Now, that copying continues in both directions until the copied DNA, those copies run into one another. Okay, the red spots, the terminus is called the terminus, where the copying ends. Now you've got two copies of that circularized DNA. Those two copies separate to opposite poles of the cell. Okay, and then that's how the DNA gets copied and separated. But now, what about for the bacterial version of cytokinesis? Well, that's called septum formation. So you see what's happening here? The center of the cell begins to grow inwards. The center of the cell begins to grow inwards. Notice how I didn't say pinch inwards. Septum formation is not an inward pinching of the cell wall. It's an inward growth of the cell wall. What's the difference? Well, remember, bacteria have a tough cell wall, right? Can a cell wall pinch? No. Just like a plant cell, the cell wall can't pinch. Here a septum forms, which means the cell wall actually doesn't pinch, it actually starts to form inwards. The cell wall is synthesized or grows inwards. And the reason it grows inwards is because of these proteins called the FITS proteins, FTS proteins. Specifically, FITZ. FITZ proteins form a ring called the FITZ ring, or the FITZ ring, or actually it's just called the Z ring. The Z ring forms around the midline of the cell, and those FITZ proteins promote septum formation, the inward growth of the cell wall until you've got two bacteria, right? And these are two genetically identical bacteria. Okay, you can see here the Z ring. That you see, this is the actual electron micrograph of bacteria, and you can see the Z ring there. Okay. In fact, is this an electron micrograph, or this actually might be a fluorescence micrograph? You don't need to know, but that ring there represents the FITZ ring, the FITZ ring. Okay, and you can see it at the midline of the cell. So I might ask you on the exam, how does binary fission occur in prokaryotes? Remember, first the DNA, the circularized DNA, has to be copied. You start at the origin of replication and copy in both directions until you hit the terminus. Those copies move to opposite poles of the cell, and then you've got septum formation. And what proteins are responsible for septum formation? You should know the FITS protein, specifically FITZ is very important for septum formation. Okay, this is the last concept on the chapter. This concept, now we're going back to eukaryotic cells, talking about eukaryotic cells again. Now, this final concept is regarding cell cycle control. Okay, cell cycle control. What does that mean? Cell cycle control refers to your cell's ability to regulate how often they divide, right? So you've got skin cells, right? How do your skin cells know when and where, what time is appropriate to divide? How do your brain cells know how often they should divide? How do your liver cells know how often they should divide? Well, it's all part of the cell cycle control system. You've got control mechanisms inside of your cell that tell the cell, that regulate how often the cell divides. And if you lose this regulation, if this becomes damaged in any way, that can result in cancer, which is basically loss of control of the cell cycle. Cells dividing far too frequently result in multiple cells where there shouldn't be. That's what a tumor is, and then tumors can metastasize, which means spread to other parts of the body. That's how you get cancer. So it's very important to understand the cell cycle control system, because that's the reason why cells know how often they should divide, and that prevents things like cancer, and it promotes things like proper development, proper growth. Okay. In fact, in your body right now, some of your cells really never divide, whereas other cells are dividing pretty much all the time. So for example, you know your spinal cord cells, those are really, really long cells that go from the brainstem and innervate through your spinal cord and then out the sides. Those spinal cord cells are not dividing. Okay, those spinal cord cells have developed, and once they've developed, that's it. They don't grow. They don't divide. Well, they don't divide. They don't grow in the sense that they don't divide. They've escaped the cell cycle. They're not in the cell cycle. They are just doing their job, which is transmitting information from the brain. Okay, and bringing back information from your sensors. Okay, But compare that to your skin cells, or compare that to your bone marrow cells, your bone marrow stem cells inside of your bones. Those cells are dividing all the time. In your hair follicles, they're dividing all the time. So some cells in your body are dividing all the time. Some cells are never dividing or hardly ever dividing. So how come my spinal cord and my heart cells don't divide, whereas my skin cells and hair follicles and hematopoietic stem cells, these stem cells in my bone marrow, How come they're dividing all the time? Well, it's all part of the cell cycle control system. So how does this cell cycle control system work? It works based on various checkpoints. It's like a clock, right? The cell cycle control system is like a clock, which has checkpoints, and these checkpoints tell the cell to stop, the cell cycle to stop dividing until a go-ahead signal is received. So these checkpoints, there's three of them, three major checkpoints that you need to know about. There's the G1S checkpoint. That's the main checkpoint. You can call that, I might even ask on the exam, which one's the main checkpoint of the three cell cycle control checkpoints? Which one's the main one where the cell actually decides to divide? And you should know that that's the G1S checkpoint, right? What does that mean? That means there's a checkpoint between G1 of interphase and S of interphase. So at the end of G1, before the DNA gets copied, there's a checkpoint called the G1S checkpoint. That's the main checkpoint where the cell decides to divide. The next checkpoint is called the G2M checkpoint. What does that mean? At the end of interphase, you've got G2, right? Well, before you enter M phase, which means before you enter mitosis, there's another checkpoint. This checkpoint makes sure that all the DNA was actually copied correctly during S phase. And then you have another checkpoint called the late metaphase checkpoint, or the spindle checkpoint. This is another checkpoint that makes sure that during metaphase, all of the sister chromatids have a microtubule attached to a kinetochore. So take a look here. Here's the cell cycle. Here's the cell cycle. This dot represents the G1S checkpoint, or the main checkpoint where the cell decides to divide. Then if the cell does decide to divide because it gets something like a growth hormone or a signal from the cell to divide, the cell will progress into S phase, copy the DNA, go to G2, and then remember at the end of G2 there's the G2M checkpoint. Remember the G2M checkpoint, that's this one. That's when the cell stops itself and double checks that the DNA was copied correctly during synthesis phase. And if the DNA was correctly replicated, you proceed into mitosis, or M phase, right? Until the spindle checkpoint. Remember metaphase checkpoint? The late metaphase spindle checkpoint, where, let me show you what I'm talking about. Look at this. Basically, there's a checkpoint here in metaphase. And what does that checkpoint check? It makes sure that every sister chromatid pair, every kinetochore is attached to a microtubule. You see this microtubule is attached to a kinetochore. Here's a microtubule attached to that kinetochore. If there's a kinetochore that's missing a microtubule, that's not good, because that could result in uneven numbers of chromosomes separating, where you could end up with both of these chromosomes going one way and 0 going the other way. That's a condition known as aneuploidy, and that is really bad. So you got to make sure that every kinetochore has a microtubule attached, so that when anaphase happens, you actually have even separation of all of the sister chromatids, right? If you didn't have the late mitosis checkpoint, then you see this late metaphase checkpoint. If you don't have this checkpoint, that could result in uneven numbers of chromosomes separating. That results in a condition known as aneuploidy, which is bad. Okay, so you could think of this control system, this cell cycle control system, almost like a roulette wheel. You've got the roulette wheel spinning, and look, once it hits one of these flaps, that's like a stop. That's a checkpoint, right? And remember, the first major checkpoint is here. This is the first major checkpoint. It's called the G1 checkpoint. It's at the end of G1 when the cell's grown, and before S, before synthesis, where the DNA is copied. There's a checkpoint there. And if this checkpoint doesn't get cleared, the cell is not going to decide to divide. The cell just remains, like, for example, if a skin cell does not clear the G1 checkpoint, the G1S checkpoint, that skin cell just stays a skin cell and does a job until it does clear that checkpoint, right? So look at this. If the G1 checkpoint clears, you're free to go and continue copying the DNA and dividing. If the G1 checkpoint does not clear, the cell will what's called exit the cell cycle. The cell stops its progression through the cell cycle. It enters what's known as G0. G0 means the cell has exited the cell cycle. So for example, again, if my skin cell does not clear the G1 checkpoint, that skin cell will just decide, okay, well, I guess I'm stuck as a skin cell. I'm not going to divide. I'm just going to do my job as a skin cell. So during G0, the cell does not have the clearance to divide, but it does have, the ability to continue doing its job. Okay. So for example, all of your spinal cord cells in the back, in your spinal cord, all of your spinal cord is in G0. Those cells are not dividing. They're not even planning to divide. They don't have any signal to divide. Humans don't even know what that signal would be to cause them to divide. But they're all in G0. So they're doing their job. They're sending and receiving signals from your brain to your body, from your body to your brain. They're sending and receiving signals all the time. They're doing their job, but they're all in G0. Okay. Now, if there was to be a signal to divide, that would clear this flap, and you would progress through the rest of the cell cycle. So G0 is like you exit the cell cycle because you did not clear the main checkpoint, the G1S checkpoint. Okay. And then remember, there's another checkpoint at the end of G2, and there's another checkpoint during late metaphase. Okay. So what happens if you mess up these checkpoints, right? Yeah, you see these little flaps? What happens if you break a flap, right? Break a flap to where it's always down. Could you imagine if you had a condition where these checkpoints were not actually doing their job, were not telling the cell to not divide? Imagine if this flap was always down. Well, then the cell would just keep going around and around and around in a circle, and you would have uncontrolled cell growth. You would have no control of the cell cycle regulation. And that's exactly what happens in cancer. You see this? In cancer, you have failure of the cell cycle control. Basically, what does that mean? These flaps are stuck down. You don't have this ability to stop the cell cycle. So the cell just keeps going into the cell cycle. One cell becomes 2, 2 cells becomes 4, 4 cells becomes 8, 8 becomes 16, and so on and so forth until you have a clump of cell. That's called a tumor. Then small pieces of that tumor can branch off and float off and land in different parts of your body. That's called metastasis and a metastatic tumor. And that's when you progress into, stage 4, late stage cancer, and that's when you succumb to cancer. So cancer is caused by loss of control of those checkpoints of the cell cycle control system. And how does that happen? Let me explain this real quick. You can see here on this slide, it tells you it's due to mutations. You know what mutation means? Mutation means taking your DNA and changing its sequence in some way. Changing your DNA sequence. That's what a mutation is, your DNA. And there's 2 types of genes. Two classes of genes, you could call them, 2 classes of genes in your body that if you mutate those genes can result in cancer. Those are tumor suppressor genes and proto-oncogenes. You see that? Tumor suppressor genes, proto-oncogenes. These are the 2 classes of genes that if you mutate them, you could very well cause cancer. Do you know why? Because these types of genes, the tumor suppressor genes and the proto-oncogenes, those are the genes in your cells. Those are the genes that code for the checkpoint proteins. You see these checkpoints? You see these flaps? What are these flaps? They're not actual flaps inside of your cell, are they? Do you know what these flaps really resemble, what they represent inside of your cells? They represent proteins. Proteins, specific proteins called tumor suppressor proteins, proteins inside of your cells, their entire job in the world, some of those proteins stop the cell cycle. They're the checkpoints. So when I say checkpoints, what are these checkpoints? What in the world am I talking about when I say checkpoints? Well, the checkpoints are specific proteins inside of your cells that tell the cell when to divide, when not to divide. Do you see? Now, where do proteins come from? Do you remember what I said about proteins? What codes for the proteins? Your genes. Remember I said that early in this chapter. I said, what's the job of a gene? A gene is simply the information to code for a protein. And it's the proteins that have jobs in the cell, right? So for example, let me give you an example. The insulin gene codes for the protein insulin. And insulin's job is to regulate your blood sugar in your body. It's a hormone, okay? But if I change the insulin gene, and that changes the insulin protein, and the insulin protein doesn't work anymore, is that going to give you cancer? No, it's not going to give you cancer because the insulin gene has nothing to do with how often your cells divide. It has to do with regulating blood sugar. Does that make sense at all? So what I'm trying to say is you can't just mutate any gene out of your 25,000 different genes in your genome. You can't just mutate any gene and result in cancer. You have to mutate specific genes to cause cancer. And those genes are the tumor suppressor genes or the proto-oncogenes. These are genes that are known to code for proteins, and those proteins' job is to actually tell the cell when to divide, when not to divide. They're the checkpoint proteins of the cell cycle control system. Does that make sense? So when someone gets cancer, the reason they got cancer is because they've got mistakes or errors in their genes, but not just any gene, genes that code for the checkpoint proteins, genes that code for the cell cycle regulation. Okay, and those genes, again, what are those called? Those are called tumor suppressor genes or proto-oncogenes. Okay, so that's how cancer works. Now you understand cancer. Cancer is caused by mutation of genes, genes that code for tumor suppressor genes, tumor suppressor genes and proto-oncogenes. Those are the genes that code for checkpoint proteins, okay, and cell cycle regulation proteins. You mess with those genes, you mess with the cell cycle control system and the checkpoints, and you mess with how often a cell divides. Cells divide too frequently. That's how you get cancer. So now you understand how you get cancer. And by the way, before I let you go, do you ever notice how it seems like pretty much anything could give you cancer? It seems, sometimes I've seen people, just, wave their hands in the air and say, oh, it seems like anything could give you cancer. So why does it seem like so many disconjointed things give you cancer? Have you ever thought about that? Like, for example, if you go out in the sun, you get UV exposure, that could give you cancer. Or if you get X-rays, too many X-rays, that could give you cancer. Or if you smoke or drink or consume carcinogenic chemicals called carcinogens, that can give you cancer. Even viruses can give you cancer. So smog can give you cancer. Well, why do all these things give you cancer? You ever wondered why all these different things give you cancer? They seem so unrelated, but they're not unrelated at all. There's actually something very common amongst all of those things. All of those things can cause mutation, which means they can change your DNA sequence. Okay, UV can cause mutation. X-rays could cause mutation. Carcinogens can cause mutation. Viruses can cause mutation and misregulation, right? So all of these things can affect your genes, and anything that causes mutation or affects your genes could potentially give you cancer because it could potentially disrupt your tumor suppressor genes or proto-oncogenes. Okay, so now you understand clearly cancer and what causes cancer. Okay, so hopefully this was an informative chapter. It is a really fun chapter to understand. Again, the better you understand the difference between sister chromatids and homologous chromosomes, the easier this chapter will be. Like I said, I got a bunch more videos for you to click on in this PDF. Click on those. Learn this material. I'm going to post chapter 11, which covers meiosis really soon. All right. Hopefully this was informative. Thank you again for watching, and tune in next time.